Hello guys, welcome to our YouTube channel, The Learning Biophysics with Team 320. So my name is Abhishek Sharma and our topic is the main biochemical process. In the biochemical process involves the decomposition of water and also the evolution of carbon dioxide and the presence of the in the environment of CO2, which is formed in the course of anaerobic oxidation makes possible photosynthesis. The bacterial photosynthesis and the photosynthesis of green plants begin about 10 to the power 9 years ago. Photosynthesis consists of the absorption of light and the transformation of its energy to the chemical energy of biochemical molecules. For the photosynthesis of for the photosynthesis to occur, light absorbing compounds were required, in particular those containing porphyrin rings, chlorophyll, and cytochromes. As a result of the absorption of light quanta in chlorophyll, the electron of the system move to higher energy levels. Then electron transport chain is operative, in which the principal participants are oxidation reduction enzymes like cytochromes. The energy stored originally in chlorophyll is released in a biological useful form in ATP and NADP also. There takes place there takes place phosphorylation evidently. At a later stage of evolution, carbon dioxide was reduced photosynthetically to carbohydrates. The hydrogen for prokaryotes is supplied by various molecules like bacterial photosynthesis, and for green plants by H2O. The oxygen is released into the surroundings. Its accumulation in the atmosphere is the result of photosynthesis. The overall reaction is 6CO2 plus 6H2O is equals to C6H12, glucose plus 6H2O, 6 molecules of H2O and 6 molecules of oxygen. About 1.5 into 2 1.5 to 2 into multiply 10 raised to the power 9 years ago, the partial pressure of oxygen in the atmosphere was 0.022 to 0.020% of the present level. These lead to aerobic metabolism, respiration. The cellular respiration involves number of interconnected processes leading to the synthesis of biological molecules required for life and charging of ATP, like oxidative phosphorylation. The molecules of nutrients are burned, oxidized to CO2 and H2O. The oxygen molecules serving as the final hydrogen acceptor. Roughly speaker, the liberation of chemical energy from food occurs in three types. The first step in the breakdown of macromolecules and fatty substances. Proteins give rise to amino acids, carbohydrates which produce hexose, and fats from glycerol and fatty acid. At the second step, these substance give rise to acetyl coenzyme A, alpha ketoglutarate, and oxaloacetate. The third stage involves Krebs cycle or CTC cycle, citric acid cycle. And the Krebs cycle begins with the reaction by oxaloacetic acid and acetyl coenzyme A. Acetic acid is then regenerated during the second revolution of the cycle. Some of the reaction involved in the cycle which perform a catalytic function are accompanied by the transfer of hydrogen atoms to NAD or flavonoid protein. For energy, two electrons transported with NADH to oxygen, there are synthesized three molecules of ATP, which is oxidative phosphorylation. These electron transport in accomplished in the respiratory chain. Upon transition from the first to the second phase, carbon dioxide is released, and transport of electrons from NADH to molecular oxygen is followed by formation of water. The general scheme of aerobic respiration is glucose plus 6 molecule of H2O plus 6 molecule of oxygen plus 36 adenosine diphosphate plus 36 phosphate is made the 6 molecule of CO2 and 12 molecule of H2O plus 36 ATP. Respiration is much more efficient than glycolysis. Upon glycolysis, the free energy change per molecule of glucose is 197 kilojoule, whereas upon respiration, it's 2880 kilojoule. The complex process of metabolism, accumulation, and consumption of energy are especially localized in cell. The respiration occurs in mitochondrial membranes, and photosynthesis takes place in the membranes of chloroplast. Biochemical processes are eventually adapted. For instance, the main source of metabolic energy for animals living in desert and for birds is glycogen rather than fats. In deserts, not only the maximum release of energy but also the maximum formation of water must be provided. The oxygen of fats produces twice as much water as the oxidation of glycogen. This is why birds have a smaller of fat. The mass of glycogen and of associated water in 8 times greater than the mass of the fat then that liberates on oxidation of same amount of energy. A detailed consideration and scheme of metabolic pathway are given in the books of biochemistry. We shall return to respiration and photosynthesis in chapter 13. Hello guys, welcome to our YouTube channel Learning Biophysics with Team 320. My name is Abhishek Sharma and our topic is strong and weak interactions. The interactions of atoms in biochemical molecules as well as in molecules of synthetic organic compound are first of all chemical covalent bonds, which we shall call strong interactions. The energy required for the rupture of carbon-carbon bond is equals to 348.6 kilojoule per mole, and the carbon-nitrogen bond energy is 336 kilojoule per mole, etc. Strong interactions that determine the chain structure of biopolymers. The binding of monometers, amino acid residues, nucleotides, hexos. Strong bonds are can be based on the quantum mechanics. The corresponding branch of physics and or theoretical chemistry known as quantum chemistry. In the course of biochemical reactions catalyzed by enzymes, there takes place a rearrangement of chemical bonds and electron shells. Biological molecules would not be able to function and also known as form of life would not be able to exist at all without non-covalent, non-chemical, weak forces operating inside biological molecules and between them in addition of strong interaction. Cell and their organelles are heterogeneous systems whose existence and functioning are determined by intramolecular interactions of non-covalent character. Proteins, which form nearly all molecules, functions in cells, interact with lipids and carbohydrates, with nucleic acid, and with small molecules. These interactions are predominantly weak, since strong interactions would have built up to rigid and stable structures devoid of molecular mobility required for biological molecules to accomplish their diverse tasks, including the delicate control of chemical reactions, compartmentation, and the setting up of concentration gradients. Let's describe the types of weak interactions operative in biological system. First of all, there are ionic bonds. The ionic bonds are the forces described by Coulomb's law. The energy of interactions between two ions with charges E1 and E2, given by like ionic bonds like E1 E2 upon epsilon R. And the second one is ion dipole interactions. This is between ions and polar groups of molecules. They are determined by the ionic charges and dipole movement of the atomic group that interacts with it. The third one is orientation forces. The electrostatic interactions between dipoles, the dipole moment P of a small molecule or atomic group is equal in order of magnitude to the product of the electronic charge 4.8 in multiplied 10 to the power minus 10 ESU by the length of chemical bond, about 10 s to the power minus 8 centimeter. And the fourth one is inductive forces. The permanent dipole of a molecule and atomic groups induces in another molecule or atomic groups a dipole moment with which it interacts. The dipole moments induced by an electric field of strength E is by P is equals to AE. And the fifth one is dispersion forces or maybe London forces. These are the interactions of the valence saturated electron shells of atoms and molecules. These are forces which are independent of the presence of charges, dipole moments, quadrupole moments, etc. Such are, for example, the forces operating between the molecules like nitrogen in the N2 nitrogen gas, in the oxygen gas, in the carbon dioxide gas or noble gases, etc. And these forces are determined for non-ideal behavior of dipole-less gases, their liquefaction. The dispersion forces are responsible for the existence of molecular crisps or quantum mechanics of origin. And the sixth one is hydrogen bonds. The specific donor accept interactions that are induced hydrogen atom in groups like oxygen, hydrogen, nitrogen, hydrogen, fluorine, hydrogen, chlorine, hydrogen, and sometimes it can be sulfur and hydrogen groups. The hydrogen atoms binds these with the valence saturated oxygen, nitrogen, and fluorine atoms. The seventh one is hydrophobic interaction. These are bipolymers, proteins, and nucleic acids function mainly in aqueous medium. The hydrophobic forces are the forces of specific repulsion between non-polar atomic groups and water molecules. They constitute the entropy effect determined by the special features of the structure of the water as the condensed formation of structures of protein, supermolecular systems like membrane, etc. That's all, guys. Thank you.